Hi, everyone. Thank you so much for joining this session. It is my greatest pleasure to introduce you the winner of the 2020 BSCB Women in Cell Biology medal. This medal is awarded every year to an outstanding female cell biologist in the early stages of her independent career. And as we all witness the negative impact that this pandemic continues to have on early career scientists, and in particular women, it's even more important that us as a community, we can support and promote our colleagues. The 2020 Women in Cell Biology medal winner is Yanlan Mao. Yanlan did her PhD at the LMB in Cambridge with Matthew Freeman, where she studied epithelial patterning in Drosophila. She then moved to the CRK London Research Institute, now the Francis Crick, where she became interested in studying mechanics and using computational modeling to study out the role of mechanical forces in regulating tissue shape in Nick Dampun's lab. In 2014, Yanlan established her group at UCL and has since continued to investigate how mechanical forces impact tissue homeostasis and repair. Since she started her lab, Yanlan earned several prizes and awards. She's a MRC Career Development Fellow, a Lister Prize winner, and also part of the EMBO Young Investigator Program, just to name a few. One of the things that for me is really fascinating about Yanlan's work is this combination of biology, physics, and computational science to address complex questions. For example, how tissue grows and regenerates. And without further ado, I will leave the stage to Yanlan.
Thank you for the introduction. First of all, I'd like to thank the BSCB for this prize. It really is an absolute honor. But this prize really isn't just for me, it's for everyone in my lab. So before we get into the science, I'd like to thank all members of my lab, past and present. It really is because of them that all of this has been possible. And they've been really incredible over the last 7 years since I started the group, making an absolute joy for the team to work together. And so thank you all for all of your efforts and in making our science environments such a joy. So what have we actually been up to in the last 7 years? Well, in my lab, we're really interested in the overall question of how tissues get to the right size and shape. And so when I took my daughter to a museum a few years ago, and I saw this display, I really had to stop and take a picture because it captures exactly what we're interested in, which is to try to understand how this huge and beautiful diversity of tissue sizes and shapes arise in nature, even though we're actually made up of very similar building blocks. Of course, we know it's all in our genes. After all, we look like our parents, our kids look like us, but how do these genes give rise to the shapes and sizes that we see in nature? And despite decades of omics research looking at gene networks, signaling networks, we're still quite far from understanding how these genes actually give rise to the sizes and shapes of tissues and organs that we see in nature. And that's because, in order to grow and to morph into the right sizes and shapes, these are all very dynamic processes. Cell growth, cell divisions, cell crawling. These are all, physical processes that really require a force to generate that motion, right? So really to therefore understand how tissues can get to the right sizes and shapes, we need to understand forces. And so the roles of mechanical forces are very important, but not just simply, pushing and pulling and shaping, but there's actually a very complex interplay between mechanical forces and genes and biochemistry of our systems. So for example, some beautiful work from the Disha lab has shown that if you were to grow mesenchymal stem cells on substrates of different stiffnesses, or else being equal, these cells will actually differentiate into different cell types and adopt different cell shapes. So this really shows the complexity of the interactions between mechanical forces and gene networks and the biochemical makeup of the systems. So in my lab over the last few years, we've been interested in 3 different aspects of how shape arises through mechanical regulations. One is how do tissues get into shape during development? Another is how do they actually stay in shape during tissue homeostasis? And then finally, how do they get back to shape if they actually are injured and damaged during the lifetime of the system? And so hopefully today I'll be able to take you through each of these and give you snapshots of some of the work we've done in the past few years on these aspects. And first of all, we'll start by looking at how tissues can get into shape during development, and we will use 3D tissue folding as an example. So many of our organs are highly folded structures. And just like this origami bird, it's very important to have these folds in the right positions in order for these tissues to function correctly. And there's been a lot of work from different labs looking at the local mechanisms and the physical mechanisms of exactly how to generate a fold. For example, apical constriction or differential growth in different layers of the tissue, or just compressive forces. But very little work has looked at how the positions of these folds are actually defined. How do these folds know where to form in the first place? And that's something we've been looking at in the lab. And to do this, we've been using the Drosophila wing imaginal disc. This is our favorite model tissue and will be used throughout this talk as the model system. I'm sure many of you have seen wing discs, probably mostly in this kind of top-down view. And as the tissue grows, it will later develop to form the adult wing. But these are the phases in the larval stages that we look at. And so if you were to cut a cross-sectional view down this axis, you will see that the tissue is actually also highly folded. And these folds are very precisely regulated. They almost always form in exactly the same places in wild-type tissues and at the same developmental times, making it an excellent model system to look at tissue folding precision. And so our central hypothesis really actually stems from work during my postdoc, where we realized that in the wing disc, there's actually differential growth patterns. So hopefully you'll see in this in silico computation model simulation that in the center of the pouch, this artificial pouch, these cells are growing and dividing faster. And that's also what happens in vivo. And as a result, the cells in the periphery of this pouch is being stretched. But this simulation is only in 2D. So in 3D, if you were to continue to do that, you would actually get the folding or wrinkling of a tissue. So I encourage you now in the audience to actually sit and stretch your t-shirts or your jumpers and see what happens. And I guarantee you'll probably see some folding of your material. And so this is really kind of the key hypothesis that we wanted to test. Perhaps in the wing disc, because of differential growth patterns, then you also get these stresses building up, and then you have folding in the precise positions that we see in vivo. So how can we test this hypothesis? And so we decided to use a computational modeling driven approach. And these, a lot of these experiments are very hard to do in vivo. And by using a modeling approach, we thought we can then test some of our ideas hopefully more quickly. And here I just like to emphasize that, in my lab, using mathematical models is really key to how we work and the philosophy of the lab. But for me, at least, modeling is not really about trying to reproduce exactly what you see. If you can already do that, then by definition, you already know everything that happens in the system. So then what's the point, of any more research work? Whereas what we use is really the aim for us is to try to simplify the system into its minimum components, taking out the essential core parts that make up the model of the system, and then use this model to really test hypotheses, make new predictions, just like an experiment in the lab. And then it's really incremental in combination with experiments that we then try to understand the system step by step. So just like this quote, all models are wrong, but some are very, very useful. And that's really the approach we take in the lab. And so we decided, therefore, to build a new 3D mechanical model of a growing epithelium. There weren't really other existing models out there at the time that would fit our needs. And so we decided to generate a new model. And so this work was very much driven by Melda, who was a very, very talented postdoc in the lab, in collaboration with Jose Munoz, a co-supervisor for this project. And so just to briefly take you through the model without going into too much technical detail. So what we do is basically taking a slab of tissue, it could be any tissue, and we discretize it into what's known as these elements. So this approach is called finite element modeling. And so each of these elements or prisms, importantly, they're not a single cell. So they could be a bunch of cells or half of a cell, just depends on how finely you mesh the prisms. And each of these prisms will be initiated with what's known as a reference shape, and this stays same throughout the lifetime of the simulation. But because we're looking at growing tissues, we also have, importantly, a growth function that can be applied to each of these elements. And this growth function comes from measured data. And so then each element will have what's known as a new ideal shape that it wants to be, okay? But then that may not be the current shape. For example, the current shape could be influenced by other elements, its neighbors pulling, stretching, compressing it. And so there will be a difference between the current shape and the ideal shape, the blue. And this difference is where the elastic energy is stored. And that's how stresses build up. And the system will want to return to its ideal ground shape. And how easily it's able to return to that shape will depend on the material properties of these elements. For example, its stiffness, so on and so forth. And so then as the system grows differentially and evolves, then you get stresses and morphogenesis building up. And so to start the model, we basically take dimensions that we see in vivo. So a small wing disc that hasn't got any folds in it yet. And then we mesh it to a degree that we know works. And then remember, the key is to try to ask, well, in this simplified system, what are the minimum sets of requirements that can induce folds in the correct positions? That's really what we are setting out to answer. And so as a sanity check, just to check that everything is behaving as it should. In this first simulation, we used uniform growth. Everything is the same uniform physical properties. There's no external resistance to the tissue. And so as you would expect, the tissue just grows infinitely as a flat structure. However, there are some prerequisites that then we have to start adding to the system to make it fold. First thing, you need some kind of external resistance, some confinement in the form of a very rough exterior resistance or viscosity, as we call it. And as you increase this, the tissue basically gets confined as it grows and it has to fold or buckle. Secondly, to get some kind of asymmetry between the top and bottom layers, we have to add the apical or the top layer, apical layer in the case of the wing disc, to make that more stiff. And then you also start to get folding that resembles a bit more of a wing disc. So for example, I'll show you a video of this highlighted simulation here. So, you start to see some folds forming. However, one important thing I'd like to highlight is that, this tissue is able to fold, but we haven't implemented any of the active forces, such as apical constriction or differential Z layers. All of the previously known mechanisms haven't been implemented yet. We can get a tissue to fold. However, of course, it still doesn't look like the real in vivo wing disc. So what else is needed? And so remember, we were thinking the central hypothesis is really about differential growth rates, right? Growth patterns. So then we had to measure this differential growth in high degree of spatial temporal detail. And to do that, what we did is to do classical lineage tracing clonal analysis. We basically sparsely label cells with GFP and then let these single cells grow over 24 hours and then count the number of cells in that clone over those 24 hours. And then from that, we can work out the number of divisions that have happened during that time. And if we did that over 3 different phases of development and also in high sort of spatial patterns, and therefore giving us grid maps of growth rates. And when we start to apply these grid maps into the model, we start to get a bit of better folding. But you see, these 3 folds are starting to form, they want to form. However, there are still issues. It still doesn't quite fold like what we see in vivo. And so what we then notice is really important is that simply by having this external resistance, sort of uniform external resistance in the model is really not enough. So what is actually providing this in vivo resistance force in the tissue? And from our knowledge, what we know is that actually the real wing disc is surrounded by an extracellular matrix known as the basement membrane. So what we've done for now, although we're looking into this in a lot more detail, is to image the matrix. And we've shown that it's very heterogeneous. So the thickness of the matrix varies, but also it does definitely increase with age. And also the patterns of the matrix does also change with age. But for now, what we've done is to just take an average value of the matrix and apply it to the exterior of the wing disc. So now you see we've got these cell elements, okay, the apical stiffness elements, the cell body elements, but also a new set of elements representing the basement membrane. So these elements, they don't have active growth themselves. They're not cells, but they have stiffnesses that we can control and they have remodeling rates that we can control. And so then as a result, as the tissue grows, they will apply different stresses to the whole system. And this is what will then generate folding in the correct places, hopefully. And so when we apply this to the model, bingo, we really see now these 3 folds starting to form in the in silico model in exactly the same places that we see in vivo. But then you may say, well, I've just tweaked, parameters to get whatever I want. And indeed, I could do that. I could probably get an elephant on the screen if I wanted to, or a mouse. So the key really with any model is to then keep all parameters the same, but change just one, okay? And so if we kept all parameters the same, but changed just the growth rates, so then we apply, for example, uniform growth throughout the tissue, what do we see? And so this is really your control. And so in this simulation, we see these uniform ripples forming throughout the tissue. So what this shows us is that, this differential growth pattern that we've used is really key for generating the precise patterns that we see in vivo. But again, any good model, like I said, should be able to make predictions, right? It's no good just reproducing what we see, but can it make predictions that we can then test in vivo? So we used the model to make several, but one prediction we did was to ask, what about if we reduce the growth in certain regions? So for example, in the central region, we know that the folds form in the central region, but their growth patterns are very complicated. And so what if we reduced everything in this region to 50%? So we made these in silico mutants where they have 50% less growth in these regions. And the model predicts that as a result, you would only get 2 folds forming, okay? Remember, the real wing has 3 folds. And so that's the prediction. So can we, and this is a fairly simple, precise prediction to test, right? And so can we then find an in vivo mutant or condition that allows us to test that? And so to cut long story short, this really wasn't easy. Many mutants are very messy, but we had to look through lots of old literature and predicted that perhaps this would work. And so there's a wingless enhancer mutant where in this key kind of hinge region where the folds form in this mutant, there's less wingless signaling, and the prediction was that it had less growth in this region, although the paper didn't show that at the time in the wing discs, but it showed the adult wing phenotype. And this is the hinge region that would result from this fold region. And you can see hopefully here that you'll see this region is smaller than the wild type control. And so we took hold of some of these mutants and quantified the growth again using the clone analysis. And compared to wild type clones, we saw that in these mutants, indeed in this kind of the hinge folded region here, the growth is significantly less. So this confirms that indeed this region has less growth. But this then result in a misfolded wing disc. And so Natalie, who then dissected these wing discs, showed, when she showed me this, honestly, I was about to cry. I was so happy. There were only 2 folds. And remember, the model predicted 2 folds compared with the wild type 3 folds. So I think this, in combination, it really shows us that these differential growth patterns are really important in generating these kind of precise folds. But of course, to then really grow these folds into the highly folded structures of the wing disc, many other forces are required and other mechanisms are required. And we're following up on some of that, and other labs are also doing the same. Okay, but so let's move on. So let's say you've spent so much effort generating your beautiful, perfect folded wing disc. So how do you then keep that in shape, throughout the rest of the lifetime or the larval lifetime? How can it buffer then other mechanical perturbations so that it stays in the right shape? And that's really something we've been asking ourselves as well. How do tissues buffer mechanical forces so that they are not constantly changing shape, constantly morphing in response to any kind of random mechanical perturbations that are always around us, of course, in real life? And so to ask some of these questions of exactly, or how do tissues cope with direct mechanical forces, we had to come up with a way where we could apply a controlled exogenous force to the system to then ask, how does the tissue respond to that force? What are the downstream reactions? So we designed this tissue stretcher, where basically your favorite tissue, in our case, it's the wing disc, but it can be adapted to any epithelia or any kind of ex vivo cultured system. And so what we do is we basically sandwich the wing disc between 2 layers of PDMS. These are elastomers that can be stretched or compressed. But then we also have micropattern these microchannels where we can basically perfuse in media, growth media, that allows the wing disc to grow and develop in this kind of ex vivo context. But also the central region is not compressed at the sides. So this is basically suspended region of tissue where we can then image and look at what happens to the system as it's being stretched or compressed. And then this whole structure, we can then mount it inside this metallic device where these arms here can move either to open to stretch the tissue or to close to compress the tissue. And this can be mounted onto a motorized device, a motor, where we can then have different frequencies, different cyclic patterns of stretch, or we can do this manually, of course. And so one of the first things we wanted to ask is, how does, for example, the cytoskeleton respond to this amongst many other things? So this little story is really just about looking at how the cytoskeleton responds. And this was driven initially by Maria and then by Natalie in the lab. And so hopefully you'll see here, this is a wing disc that's being clamped. And this is the central region that we image. So we can do this through live imaging, and that's what we do through all of our data. And this is an example of a stretched wing disc. Hopefully you can see this region here has been stretched in this horizontal axis. Okay, so how does this tissue respond? And one of the most striking things that Maria saw when she did this is that when we imaged myosin 2, normally it's quite homogeneous around the cortex of each cells. So this is ekaterin, the control, where you can see the outlines of all the cells. But when we stretch the tissue, within a few minutes, the actomyosin polarizes to form these supracellular myosin cables, whereas the ekaterin control doesn't really do that. And so to cut a long story short, what we found is that this really changes the mechanical properties of the wing disc. So if you don't have this, or if you don't allow this to happen, for example, by making clones where there is no myosin, what happens is that when you stretch the tissue, this region where these myosin cables cannot form, the cells are much softer. They stretch much more, they deform much more, and this can be quantified. And also, when you relax the stretch, normally the wild type cells would go back to their original shape. However, these mutant cells that cannot form these myosin cables, they do not go back to their normal shape again. So basically these actomyosin cables, they stiffen the tissue and they also allow it to be more elastic. And so without them, the tissue is much softer, much more deformable, and also less elastic. All right. And so molecularly, we were able to show that this whole process is actually dependent on diaphanous, which is an actin, which is a formin. And so that is responsible for actin polymerization. And so this then allows these myosin cables to form, to then prevent the tissue from being deformed. But if you don't have this, then upon stretch, whether it's intrinsically generated or externally generated, then the cells will deform. And actually, we show that this really affects the shape of the wing disc as it's developing. And so this was published a while ago, so hence, very brief summary of the story. But I think what it shows is that, there are different mechanisms in which, tissues can cope with mechanical forces or perturbations. And one mechanism is to polarize your actomyosin structures, for example, in this case, myosin, which then changes the material properties of the tissue, preventing it from being deformed and therefore preserving the shape of the tissue. And so, however, what about if, the whole structure, let's say the forces are so high that it actually damages the tissue, inducing, injury, a cut, or, basically loss of limbs or structures. So, for example, like this poor salamander, if you were to cut its limbs off again and again, it will actually regenerate its limbs and again. So that's, brilliant for the salamander, but most of us, including as humans, are really not very good at doing that. And so we need to have better, improved ways to try to repair our damaged structures, whether it's the skin or the liver or the gut. And so how can we do that in a more precise way? And so mechanics is also very important for that, we've shown, and this is a little story about that. So this part of the work has really been driven by Rob, who's a very talented postdoc in the lab. And what he's been doing is to set up different winding assays, so very precise winding assays to wind the wing disc. In this case, he uses laser ablation to precisely cut around 10 to 15 cells every time from the wing disc epithelia. So this is the wing disc again, a top-down view. And what he's been doing then is to use automatic image segmentation and tracking softwares that we developed in the lab, amongst other softwares, to then track the dynamics of this repair process to then understand what actually happens as the tissue heals or tries to heal itself. So there are several things that occurs upon winding. So for example, this would be the epithelia before winding. And upon winding, you'll see this high recoil. So basically the wound expands very quickly. And that's really the release of tension in the system. And then very quickly, within a few minutes, you'll see an actomyosin purse string forming around the wound. And these actomyosin purse strings are being shown in other structures as well. And so as a result of this, you'll see rapid, fast closure of the wound, this very fast phase that you see here. And then, however, there's also a slow closure phase here, which then completely closes the wound to a single point. And so this slow phase and fast phase really have very different dynamics. So we thought perhaps, very different mechanisms are happening in these phases. So what is really happening in the second phase? So we know purse string drives the first phase, but what about the second slow phase of wound closure? And so Rob looked at many, many things, but one thing that was really striking, and I'll show this in this movie as I play it later, but I just want to highlight you to look out for what I'm going to show you. And so basically this would be a typical wound. The white area is the gap, the wound area. And then these blue cells are the existing or the remaining cells surrounding the wound edge. And you'll see that they're all initially surrounding the wound edge. Okay, they all share an edge with the wound. But as I play the movie, watch out for these dark blue cells. They will start to leave the wound edge one by one through process of cell intercalation, whereas the light blue cells remain at the wound edge. Okay. So hopefully you could see that. So now you only have light blue cells at the wound edge. And so these stills also might make it easier. So dark blue surrounding, neighboring the wound edge, and then it's being squeezed out or intercalated so that eventually it no longer shares an edge with the wound. Okay. And so these events, what we call these intercalation events, really occur at exactly the same time as the onset of the slow phase of wound closure. And these, what we call intercalation events, are also much higher in this wounded tissue than an unwounded wild type, steady state tissue. And so this made us kind of hypothesize that perhaps these intercalation events are really important for driving this sort of slow phase of wound closure. But is that really the case? How can we test this hypothesis? And so, what is the, really the function of these intercalations? And so one thing we noticed is that, in fact, before we saw these intercalations, what happens to most wounds when they heal is that, for example, in the embryo, in the Drosophila embryo, what happens is, as the wound closes, all of the cells at the wound edge, they change shape, they taper into these long elongated cells to close the wound gap and form what's known as these multicellular rosettes. None of these cells will intercalate or move away from the wound edge, but instead they just change shape to meet in the center. Whereas in our system, what we see is that, in fact, after the wound closes, there's no obvious sort of patterning changes. In fact, the cells preserve their original patterning. The cells do not have this elongated shape, basically don't form a scar. And so this leads to very seamless wound healing. So perhaps this is the role of these intercalations. So not only does it drive faster closure, but also perhaps leads to more seamless wound closure. And so to test this, again, we decided to use initially a computational modeling driven approach. And this time we collaborated with Mike and Sheila in the Department of Physics at UCL. And we used a traditional, classical 2D vertex model. So I won't go into the model in detail. It's a fairly old model. We didn't really develop it. But what we used it for is to use it to adapt the model so that we can look at wound closure. So for example, one key feature we had to add was that at the wound edge, for example, it's a typical simulation here, these wound edge junctions would have higher contractility. So there is a contractility term in the model. And so by increasing this contractility, we're able to simulate the lactomyosin press string. Also, in the model, we have to allow these intercalation events, so neighborhood exchanges, so that they're not stuck in multicellular rosettes. Okay, by having these, this would be a typical simulation. Okay, so then we wanted to use this again, like I said, to be able to test, compare simulations by changing one feature and versus another to look at what happens to the in silico wound closure dynamics. And so, for example, one thing we want to test was, these intercalation rates. Remember, what we're asking is, what are these intercalations doing? So in this simulation here, this patch of tissue is able to undergo very low rates of intercalation, whereas this one is able to undergo higher rates of intercalation. And as a result, hopefully you'll notice that this wound is closing much faster than this one. And Mike reassures me that he didn't just play this video faster than this video. And I of course, I believe him, but also you can see that clearly this one, even when it eventually closes, it closes very differently in pattern to this one, right? So you still here, you can see lots of cells neighboring the wound edge, tapering in, whereas this one is much more seamless in its closure. So therefore, two things the model is predicting is that this, these, by having fast intercalations, or what we call a more fluid tissue, you actually get a higher rate of wound edge contractions, and this leads to faster wound closure, but also it leads to more precise wound closure patterns. And can we test that, therefore, in vivo? Is there a way to change the fluidity of a tissue, i.e. its ability to intercalate, and look at what happens? So genetically, there are different ways, but one way we did to decrease fluidity was to increase myosin activity in the system by knocking down MBS. And conversely, to increase fluidity, we decrease myosin activity by knocking down Rho kinase. And so when we did that to hyperactivate myosin, remember, this solidifies the tissue more. And we did the wounding assays again. What we saw is that basically the wound gets stuck. After a certain extent of closure, it's no longer able to close. Okay? Whereas if we increase fluidity in the system, then what happens is initially it's about the same, but then very quickly it's able to intercalate like a much more fluid tissue. The molecules, or the cells in this case, are just flowing into the wound edge to close it in a much faster rate. And quantifications show that this more fluid tissue actually closes in half the time of a wild-type tissue. And of course, it's able to undergo intercalations much faster. So I think, again, what we've shown is that both through modeling and through in vivo experiments, we've been able to confirm that these wound edge intercalations are actually really important in driving wound closure, and also preserves the cell shape after wound closure. And even if you reduce myosin activity, for example, although you would reduce the purse string strength, but because you're in this case fluidizing the tissue, it can actually overcome even a weakened purse string. And this more fluid tissue literally just flows into the gap and seals the wound much more efficiently. So this, I think, again, really shows that the material properties of a tissue can really help its wound closure dynamics. So finally, in the last few minutes, I'd like to show you some preliminary data of unpublished work, which Rob is continuing to do, because we've noticed that, again, the wing disc, it's not a 2D structure, right? Like you saw in the first part of the talk, it's a highly 3D structure. And the same applies to the wounds. You have these wounds that are spanning the whole length of the epithelia, or the height of the epithelia. So how does this 3D wound actually heal itself? And one thing Rob has noticed is that as the wound heals, not only does it undergo these 2D intercalations, but also the apical surface indents or sort of curves down. And same for the basal surface. The basal surface, curves up. And so these indentations in 3D actually basically grow as the wound closes. And so this then led us to another hypothesis, which is that actually perhaps these 3D cell shape changes. So initially these long tall cells because of after wounding, they change shape, they shorten, and perhaps just by shortening, if volume is preserved, which it seems to be, then it's able to increase its XY surface area. And this may be sufficient to actually close the wound temporarily. And then other forces will come into play to then, regain the height of the epithelium. So this is kind of the hypothesis we're working on. And one prediction from that would be, the bigger the wound, then the deeper these indentations have to be. And indeed, that's the case. That seems to be what we see. If you have a bigger wound, then you get deeper indentations. And so a few remaining questions that we're addressing now is to understand, well, what drives these apical basal shortening of the cells? And do these 3D cell shape changes actually contribute to wound closure? So what Rob is saying is that if you were to look, more basally down this tall epithelium, you actually see these, what we call lateral myosin cables, stretching the sides of the cells, spanning the sides of the cells. And what we predict is that what we think these cables are doing is that they're actually contracting the size, shortening the cell. So to test this, and to test whether these cables are actually under tension, Rob did laser ablation to cut these cables. And what we see is that if we track the starting position in green, after we cut, then the apical regions of the cells, they actually rise again. So you'll see this basically this kind of recoil of the apical surface, which shows that the system is under tension indeed. But also, when Rob did these cuts and we track the apical area of the wounds, the area also starts to increase again after we cut these lateral cables. So these cables also seem to be stabilizing their area reduction. And so what we think is happening is that there's a complex interaction between the lateral sides and the apical surface to drive much more efficient wound closure. And these help to stabilize the wound as it's closing. And so finally, as I'm running out of time, I'm sorry, really in summary, what I've shown is that there are many, many multiple ways in which a tissue can cope with different types of mechanical forces. During development, many of these forces are very much instrumental. They really help to shape the tissue in 2D and 3D into its optimal shape as it develops and grows. But also, there are mechanisms that also help to buffer these forces. So for example, if there's a temporary perturbation, temporary stretch, mechanisms also kick in to prevent the tissue from being constantly morphed and shaped in response to any mechanical fluctuations. And so altogether, this really determines, a way in which a tissue can grow to its right size, its right shape, but also maintain this shape during tissue homeostasis. And after wounding, it's able to repair itself to get back into the right shape again. However, even in the wing disc, we've seen there are lots of different mechanisms this tissue seems to use to cope with these different forces. And how does it know which one to use upon different types of perturbations? This is really something we're interested in now in the future, to try to understand the choice of response under different circumstances. And so I'm very much looking forward to working on that in the next few years, and also testing some of these ideas in different systems and different tissues. And so finally, I'd like to thank everyone again in my lab. So I think I've highlighted all the work of the different people. I've mentioned them throughout the talk, but also lots of people in the lab now that I couldn't talk about their work. There just wasn't enough time. And obviously during COVID times, the best we could do recently is to meet via Zoom like we're doing now. It's a shame that we have to meet in this format, but it's better than nothing. And hopefully we'll be able to see everyone in person very soon. But also I'd like to thank my collaborators and also everyone at the LMCB for all of their support over the last 7 years. And it's been really great to work in such a supportive environment with such terrific people. And this prize is really for all of them. Thank you for your attention.
Thank you so much, Yanlan, for your amazing talk. Before we go for the questions, I would like just to share your medal with you. We cannot do that in person this year, but we will share it virtually. And here he is, the Women in Cell Biology medal, which you will receive in person by mail.
Thank you very much.
Now we go for some questions. I think there are some questions already on the website. So one question is, do you have a global map of ECM thickness across the wing disc? Is the ECM thicker on the central pouch region, for example?
Right, yes. So as I said, we are starting to do that, and we have some preliminary data using EM and also other imaging techniques, which suggest that indeed the pouch region seems to be the thickest. And as I said, also this changes with age, but generally it starts out pretty much the same thickness throughout. But as it ages, the pouch region gets thicker and thicker, whereas the other regions, for example, the hinge and the notum, seems to stay kind of the same. So indeed, yes. And so hopefully we'll quantify that soon, because again, it needs lots of quantitative data and to be sure of that.
So another question is, what are the external forces acting on the wing disc in vivo?
Yeah, so that's a really interesting question. That's something we're starting to again quantify through imaging and analysis right now. So just so that for those who don't know, normally the wing discs are kind of embedded in the larvae. It's kind of attached to different tissues, and it's kind of swimming, but not completely loosely swimming in the larvae. So as the larvae move, so like peristalsis, the larvae are contracting and crawling, moving forwards, backwards, wiggling, turning, as it tries to get through food and so on. So that's actually, there's a lot of forces going on there. So what we've been doing with the new postdoc in the lab is to image this through the larvae as it's doing these peristalsis movements. And already we have some prelim data where we can see the wing disc getting stretched sometimes, getting, sort of stretched and then relaxed. And as it's moving and crawling, it's really pulsing, often sometimes, a few milliseconds, sometimes a few seconds in duration. So clearly there's a very dynamic range of forces or strain and strain rates that it would normally experience in different environments, different confinement. And so again, we're quantifying this because I think that will be really interesting to really understand how the tissue buffers these, and when a force becomes too much, what are the physiological ranges, and when we then apply different sort of exogenous stretches, what's within physiological range, what's without, what's outside of this range. So that's all kind of very interesting. But clearly it's not static. It's definitely undergoing lots of strains and stresses as it's even during normal development.
During wound healing, the livers and remaining cells are dependent on the wing disc orientation?
So I missed that. So during the wound healing part, right?
Yeah,
so,
the wound, yes.
Right, yeah. So not really on orientation of the wing disc itself per se. It really depends on the kind of the pattern of the wound, right? So relative to what's left. So although we try to be as controlled as we are, but normally the epithelial packing in the wing disc is quite a kind of distribution of polygons. It's not always, for example, perfect hexagon. So even though when Rob tries to use the same kind of region and tries to pick some number of cells, the exact wound shape you create is never exactly the same. And so, for example, you'll leave sometimes, pattern of like 10 cells surrounding. So that pattern, that initial pattern of what's left, surviving cells that's left, that's probably what's determining who goes first. Because basically what happens is every cell will then, at the wound edge, will then start to shorten this junction. And it's always, by definition, the ones who shorten first will be the ones that that leaves the wound edge first, and whoever shortens first will really just depend on the pattern that you get when the wound is created, not necessarily on the pattern and orientation of the wing disc. However, having said that, there are of course some global patterns even in the wing disc, and so that will also, of course, if the question is more about that, then I guess that global pattern will of course also affect the pattern that you create when you're making the wound, depending on where you aim your laser blasting laser. But again, we haven't really done that. We've really focused for now on fairly sort of central regions, not on the boundaries where the patterns are not too irregular, because again, that's another question, another variable. We're in a very variable system already, and for now we're trying to focus on one thing at a time.
We just have time for one more question. What about if you induce wing disc overgrowth? Does your model predict the same changes that will occur in the tissue?
Yes, indeed. So to some extent. So of course, if it's really extreme overgrowth, then it gets very complicated, and you get sort of tumor-like growths happening. But what we've done is, and it's published in the paper, we've induced localized overgrowth clones, where it's fairly controlled but clearly overgrowing. And what happens in vivo is you often get a ring of folding around the clone, so around the kind of overgrowing and normal boundary. And the model predicts exactly that as well. So you often get an indentation, the fold starting to initiate, at this high growth versus normal growth boundary. And we see the same in vivo as well. But one has to be careful with lots of these, more extreme, because then the whole thing becomes very messed up and you can't really predict much. Yeah.
Many thanks, Yanlan. We are out of time basically, but there were more questions to be asked. So I think the best is for people to join Yanlan on Thursday evening, isn't it? You are hosting
that's right.
breakout room sessions, so people should join and ask more questions.
Sure, no problem. Or feel free to email me or whatever, chat on this. I think there's a chat platform on the conference platform as well. So
yeah.
Okay, thank you very much.
Thank you. And just before everyone leaves, I just want to take this opportunity to thank all the speakers and all of you for joining today. And we hope you enjoyed these sessions, which have been really outstanding. And we look forward for more exciting science tomorrow. And there will be now 2 social events. One is a roundtable to discuss the transition to independence. And this will be hosted by myself, Inej Han, and Sarah Ulner. So join us if you want to know more about it. And also Rowan and Alex will be hosting a fun pub quiz. So my suggestion is grab a drink and join us for these social events. And hopefully see you later.